Hello everyone, I'm Cassie, Marketing Specialist from GenScript. So just to give a background of GenScript, we are the leading biotech company providing life science services and products such as gene synthesis, peptide, protein, antibody, and clinical drug development service. Today's topic for the webinar session is COVID-19 and emerging bat zoonotic viruses in a quarter of century. And I'm very excited to introduce you the presenter today, Professor Wang Lingfa. He is the director of the program in emerging infectious diseases at Duke NUS Medical School, Singapore. His research focuses on bat-borne viruses to better understand virus and bat interaction. He holds a number of honorary positions and memberships and has received awards such as the 2014 Eureka Prize for Research in Infectious Diseases. This webinar will take around 30 to 40 minutes. If you have any questions, please feel free to type in the questions field that you can see on your screen, and we'll be answering them after the presentation. If we didn't get to answer your questions during the Q&A session, we'll be sure to take down and we'll email back to you. We'll record this webinar and it will be available on demand on our website after today. And lastly, here's the list of the upcoming webinar that we are going to be rolling out in the following weeks. So without further ado, let's welcome Professor Wang to start his presentation. Professor Wang, over to you right now.
Okay, thank you. All right, let's see, I put on the slideshow mode. Can you see the full screen now?
Yes, I can.
Okay, so first of all, thanks for GenScript for the opportunity, and thanks all the audience. Good morning, good night, good evening. Depends where you are, okay? Yeah, so I thought, COVID-19 is in the news every day in terms of, transmissions and asymptomatic infections. So I thought I will focus on today's topic. It's more into the area of my research. It's really sort of put COVID-19 in the context of emerging bat-borne zoonotic viruses. And this, I've been doing this exactly for 25 years, so it's a quarter of century. So here's the outline, okay? So briefly, we will go through, quite a few topics, okay? And I will give a very quick review, basically. Obviously, this is not the only emerging zoonotic virus of confirmed or suspected bat origin. Importantly is, the world is caught by surprise of this COVID-19, but for scientists like myself, it's totally within our prediction, events like that. But I have to say the scale of this outbreak still exceeded our expectation. And then I will give you a little bit of a rundown of my personal involvement in this COVID-19 response from internationally to nationally, and the lessons we learned, and I wrote a comment in Lancet, and I will summarize a few of the points there. And then early events of the COVID-19 outbreak, for outbreak like this, it's really important to intervene early. So there's lessons there. And then the virus origin and also the early transmission events. And what's next, in terms of my own research and the field. But I will finish with, the topic of bats, because it's in the media, some try to demonize bats, and I always say it's not bat's fault. So it's a friend or foe, Okay, let's go to the next slide. Oops, sorry, it's why it's jammed. Hello. Hello.
Hello. Yes.
My slide. Okay, my slides is a little bit slower. Okay. Yeah. Okay. All right. So this is a brief summary of the major emerging zoonotic disease events. So I caught from Hangzhou to Wuhan. So the first virus that I named and discovered is called Hangzhou virus, 94 in Australia. It's a bat-borne virus, and then it spill over to horses, get an intermediate host horse, eventually get into human, and the mortality is like 60%. And the next one is Nipah virus, near where we are, Singapore, Malaysia, so discovered in Malaysia. But now it's also in Bangladesh, in India, Philippines. So this is a virus jumped from bats and amplified in pigs, and the mortality can go to 70% to 100%. And then I think with COVID-19 now it's called a SARS-2. So we're all familiar that 17 years ago we had a SARS. So I led the international consortium to discover that SARS is also a backbone virus and the CIVIDS is the intermediate host. And then we have the Middle East respiratory syndrome virus MERS 2012, start from Saudi Arabia. Camels are now the intermediate or the second reservoir host. 2014, we had a major Ebola outbreak in the West Africa, and this one is most likely a direct back to human transmission. And then 2019, we had a COVID-19. So right now, I think we believe it's a backbone virus, but in terms of intermediate host, it's still unknown. For some reason, the slides, okay. So ever since the discovery of the vast genetic diversity, so we know there's lots of SARS-related coronaviruses. Now, this is a species in the taxonomy of coronavirus. And we know that bats all over the world carry this kind of virus, and there's serological evidence that human exposure to this kind of virus. So we have been predicting to say, SARS-like virus will, reappear. But we were only sort of half right because, this is a family tree of the bat coronavirus. That's like, 10 years ago. So we said the one that human, the coronavirus, as you can see, is this one, in red. So when people ask me the question, where SARS can come back, SARS is here. So the question is, will SARS come back? And I said, yes, it will definitely come back. But when MERS really emerged, it still caught us by surprise because it's not really closely related to SARS, and it's in a different branch of the beta coronavirus. So we modify our prediction, say, there will be more outbreaks caused by bat coronavirus. It could be SARS related, it could be MERS related, it could be even not related, but it's a bat coronavirus. Okay, so 2013, after the MERS, excuse me, outbreak, I was interviewed by the Straits Times in Singapore, and I made a sort of a scientific prediction. I said, I'm almost certain in the next 10 years that a new killer virus spread by bats will emerge. So that's 2013, and sadly, I think I got this right. COVID-19. 66 years later, we got COVID-19. So again, for people working in this field, we have been working to prepare for this. So this is a grant, I applied and got, in 2016. This is a National Research Foundation in Singapore. So it's a Singapore-China collaboration with the National Science Foundation in China. And look at the title, it's combating the next SARS or most like emerging infectious disease outbreak by improving active surveillance. Okay, so during this sort of a grant period, we had another coronavirus outbreak in China, which killed 25 piglets. And then that's in 2017. And now 2019, we have a coronavirus that, kills human. So last year, I also put a review in the current opinion in virology. And as you can see that I sort of predicted to say that, in fighting outbreaks, it's like a fighting terrorism, Donald Rumsfeld had the terminology of you have the unknown unknowns, the known unknowns, and the known knowns. So bat coronavirus is the known unknowns. We know it will jump, but we don't know which one will jump. But I predicted that bat coronavirus will be the more likely cause of future spillover into humans because the genetic diversity and past experience. Okay, so now we come to what I have been involved in this COVID-19 sort of outbreak response. That I was fortunate enough, somebody in WHO observed, said I'm the only non-retired EID scientist because many of the EID scientists in the Hendra and the Nipah outbreak are already retired. So I was old enough to be involved in Hendra and young enough to still practicing in the COVID-19. So the other thing is interesting because I had a long-term collaboration with the Wuhan Institute of Virology. So I had a trip planned in January and I was on ground zero, sort of speaking, right? But that trip was almost by accident. I did not go there to investigate COVID-19 because it was pre-arranged, but I still get a personal experience on the ground. And when I came back to Singapore, before the Singapore government, started this quarantine policy, I self-quarantined myself because, as a virologist, infectious disease person, I know that how risky it is. And then, I had a 17-day quarantine, and in these 17 days gave me a lot of opportunity to read a lot of things. And then I served on multiple WHO OIE. OIE is the World sort of Animal Health Committees, including that important committee on the WHO IHR Emergency Committee, which declared, the emergency of public health emergency of international concern. And then also in Singapore, I'm on the national team in charge of the virology and serology. And last but not least, I think, this is important, right? COVID-19 starts from China, and during quarantine, there's so much going on in the Chinese official social media. So that happened to be the time I was quarantined myself. So I had the opportunity to read both English and Chinese media, and that, I think, put me in a very unique position. Not only that I have involved in the 5 major emerging zoonotic disease outbreak response, but also in this one, COVID-19, I think it's really important to read the mode and also the reports of both English and Chinese media. Okay, so I just want to show some photos to demonstrate, the difference, right? January 18th is the day I left Wuhan. So I took a direct flight from Wuhan to Singapore. And the photo on the left is that night before boarding. It's as usual, right? Any gate of airport, when you're board, it's crowded. People are lined up, try to get onto airplane first, And noticed nobody wear mask, 0%. And then few days later, basically, 31st of January, that's the last direct flight from Wuhan to Singapore. And now 100% people wear a mask. So what's the difference? I mean, there's 2 differences. One is on January 20th, the Chinese authority declared that the virus is capable of human-to-human transmission. And also January 23rd, the government decided to shut down the city of Wuhan. So these are some photos, during self-quarantine, I was able to do teleconference with WHO, and I was, of course, still working on some papers and reports. And I managed to publish a quite important commentary on Lancet. So the title is From Hangzhou to Wuhan and What Has Been Learned in Responding to Emerging Zoonotic Virus. So I'm not going to go through all of them, but I think the number one is very important. I will touch on later is that how do we report, a disease outbreak like this at the early stage? Because Cox postulates this is the very strict conditions that you confirm there's a new pathogen, that you have to be isolated the pathogen from, patients, and you be able to purify the pathogen, grow in vitro, and then put back in healthy people and reproduce disease. So that was 100 years ago, Obviously, in the modern era of next generation sequencing, we can do much better. But we don't have a policy yet from WHO to national policy is, can we use next-gen sequencing data to report a new outbreak? The second point is the lesson now, both for the Chinese scientists and for us, outside China, is that as scientists and clinicians, for our career, there's a major KPI, performance indicator is publication. But during an outbreak, how do you balance outbreak response, saving lives, and then do science and publish, right? And then the third one is that this is a disease mainly affect human and severe and death, but it's emerging zoonotic virus. It originated from animal, although we still don't know what the intermediate animal is. So the approach has to be a one health, right? Not only doctors, but vets and zoologists, environmental scientists need to be involved. And the last point I will come back is that naming a disease or virus nowadays is as difficult as almost discover the disease. So what I learned from COVID-19 is that social media is really a double-edged sword, right? It's very important for information, but also there's so much fake news. Even some of the scientists are really comment, very loosely, and it's not really good for the society. And I always say it's not bats' fault. Okay, bats has been around for longer than us, and I will come back as the last point. I will discuss why that's the case. And then the other lessons is that, as I said, you're working on fighting emerging infectious diseases like COVID-19 is no difference like fighting, terrorism or a military war. So this is really, we have to do the training and the infrastructure building and networking during peacetime so that we can win the battle during war. This is just a quick sort of a review of the major events, and WHO was alerted on December 31st, and then on January 9th, WHO stated that the outbreak is most likely caused by previous unknown type of coronavirus. And January 21st, so the 20th they had a media conference in Beijing, and 21st the Chinese authority officially declared there's human-to-human transmission on 21st. But these are the kind of the early events most people know in the public domain. But to me, as a scientist, I think there are much more important early events that are not really well publicized, right? So now some of them, we only learn this retrospectively. For example, the first confirmed cases of this severe pneumonia in Wuhan is on 1st of December. So that's like 1 month earlier than the public knew about that. And most importantly and impressively, that the doctors in Wuhan, they actually noticed that, and they sent, lung lavage samples to private sequencing companies, and they had the data suggest bat SARS-like coronavirus on 26th of December. And now the next point is sort of circulating in the media, right? The 8 doctors start to have a WeChat group and say there's a confirmed SARS case in the hospital in Wuhan, and then these people got into trouble, and then later was sort of rectified. Live virus was already isolated on January 5th, in the Wuhan Institute of Virology, and the first genome sequence was obtained, during early January, and was shared in a scientific sort of database GSET on January 9th, 10th, and then WHO got a sequence on January 12th. So imagine that if we responded according to these early events, rather than the one we learned from public, I think we will be in a very different sort of game right now. Okay, and so the difficulty, as I said, is that do you go for the pathogen and purify and do the classic Cox postulate study and then announce to the world and WHO, or you announce early and using next-gen sequencing results? There was lessons, so that's why, to be fair to the Chinese scientists and authorities, during the SARS outbreak, very similar to the COVID-19, happens in winter, Chinese New Year, and then the etiology, in those days, we don't have next-gen sequencing, so rely on classical virus culture. So there was 2 camps. One says there's a virus, but they could not pinpoint on which virus, but they could not isolate. The other camp says that under EM from the lung samples of patients, they can see chlamydia, a bacteria. So the Minister of China basically leaning towards the chlamydia theory and announced to say, have a happy Chinese New Year because we found the etiology is a chlamydia and we can use antibiotics, so no worries. And of course, he got it wrong, right? So then the cases keep coming up and peaked around March, April in 2003. And the Minister of Health in China lost his job because of that. So you can imagine, early 2020, if you are authority people in China, you are not going to, commit the same mistake. That's why I think the cautious reporting system was slower than we liked. And then you can, interpret this as a slow response, or some, media, even accuse this of cover up. As a scientist, I feel like, you have to give them the benefit of doubt because they have the lesson for SARS. If you report too early, you may got wrong. So my question is, should we continue to report confirmed etiology? That means you have to isolate the virus and confirm, or should we now encourage clinicians and scientists to report next generation sequencing data from suspected etiology? So you can declare, I'm only report suspected etiology and that need further confirmation. I mean, there's a lot of plus, but the minus is that if you get these 2 rules, then you get red herrings, right? Distractions all the time, then people will be reporting contaminated sequence or commensal sequence. So this is a tough decision, and it has to be made at a high level from WHO and to national bodies. So I think personally, I'm pushing for this, but I don't know when we can do that, how, and we can do that. Okay, so in terms of the the viruses, there's lots of debate about is that a bad virus and how many different viruses are circulating. So this was an early paper that conducted by bioinformaticians, and some of the data is interesting. Basically to say from the original sample, and now we have like lots of genome sequences. And if you plot this genome sequence according to time and also geographic locations, like from Australia, England, USA, and that's where the initial discussion comes to say, oh, the USA actually have more, variants than, other parts of the world, so maybe the virus start from USA. I think that's an over-interpretation of this data. What this data really says is this paper is that to say they have evidence now, the first known case is December 1st, had no market link. Remember that Huanan seafood wholesale market. Initially, we thought that's the place that the virus started from, animal to human transmission and followed by human to human in the community. So this paper basically says that's not the case because that the first confirmed case had no market link, and even the first batch, 41 cases, 13 of them had no market link. So what they concluded is that the market may not be the original source, but definitely play a role in amplifying the transmission. So that really indicates that whatever is sold in the market, some animals are susceptible and can amplify the virus. Another important conclusion from this paper, which is supported also in the WHO report, is that the virus is genetically more stable than SARS or MERS. That can mean both good and bad, right? Because for the general audience, when you think the virus mutate, it's all bad. But actually, that it could mean that the virus becomes less virulent because the virus does not want to kill the host. Viruses really want to evolve so that they can permanently co-live with humans. Okay, so that's actually, it's not bad news. The bad news is if they change it too rapidly, then the vaccine and the diagnostics that we're developing may not be applicable to all the variants in future. Okay. The last point is also important from this paper is that through the phylogenetic analysis and molecular tracing, they conclude that the first animal to human transmission may have happened in November or earlier. So this is now more and more people sort of believe that's the case because the second last point to say the virus is genetically more stable than SARS or MERS is that when you have an animal origin virus transmit to human, usually early phase, they will adapt and they will mutate. So when you get into a genetically stable form, usually means that the virus has been in the new host, in this case human, for some time. So these 2 points are sort of interlinked. The next slide I want to show you is the lockdown, right? There has been so much debate last month, at least 2 to 3 weeks ago. I mean, UK government even says we're not going to go for lockdown, we're going to go for herd immunity. Of course, now they changed their mind. So this is the curve of newly identified cases in China. As you can see, it really peaked in early February, right? And then they start to really report on January 20th, after the human-to-human transmission. There's one shining example, most people really may not know that there's a city we call a small city, it's a 1 million residents. In China, 1 million is small. Wuhan has 11 million. So this city is called a Chengjiang city, which is outside of Wuhan and around 100 kilometers from Wuhan. Obviously, that it's under really high risk, part of Hubei province. So remember, the Wuhan government shut down the city on January 23rd. But this mayor from Xinjiang city, he made a brave decision to shut down the city, lock down the city on January 17th, 1 week before Wuhan. And now you look at all these cases in the Hubei province. So I highlight in red. And for those who could not read Chinese, I apologize this, So 185 cases is in this city, Xinjiang city. And then let's compare another city with similar population and similar geographic locations. is the Ezhou city. So they had 1,339 cases. The only difference is that one city locked down 1 week before the other city, and what you get is 8-fold difference of people infected in that city. So I think the Chinese already have the data to suggest that really lockdown is not only effective, but maybe it's the only way to control this outbreak. So now I move to the topic on the origin of early transmission events. As I said, this is really my area of research. And so the group in Wuhan Institute of Virology, I collaborate with them very, very closely. And then Professor Zhou Peng, who was the first author of the Nature paper, was my former PhD student in Australia and a postdoc in Singapore. So from their data, it's very clear that now, among all the viruses known, means that we have the sequence, that the bat coronavirus is the closest to COVID-19 virus. It shares 96% genetic identity, and that's very, very significant. I will come back later to say why that's significant. But as I said, my personal experience, we discovered the Hendra virus in Australia, and then later we discovered that bat is the reservoir, natural reservoir. You need a horse and transmit to human. And then the SARS, you need a bat to civet to human. And the SARS-CoV-2 or COVID-19 now, we think is a bat virus, but the animal X, which is the intermediate animal, is still missing. And you must have read in the media in the early stage, people suggest snake, and I think it's unlikely. And now I think everybody is pointing to pangolin. I think my view is possible, but we need a much more conclusive scientific data. So the flavor of the month is sort of pangolin. They have papers and the papers and the papers on pangolin, and they even have a Nature paper just published last week. So this is really the main conclusion of paper is they say, really all the studies are based on sequencing, and one had a very simple ELISA data, and showing only 1 out of 8 has a weak positive. So that's something I'm not too happy. And as I'll come back later, I'm a strong believer that you need a serology data. And overall, the genome sequence is around 90%. So it's lower than the bat coronavirus. But the key domain, which is called the receptor binding domain, which is the virus component that interact with the host cell, is more related to COVID-19 than bat coronavirus. So the hypothesis now is that COVID-19 or the SARS-CoV-2 is a recombination between the bat and pangolin coronavirus. And I personally think that that's too early to have that kind of conclusion. All right, so this is like a menu. And again, for those who could not read the Chinese, I apologize. So this is the real menu, okay? Somebody took a photo before the Huanan market was shut down. It's incredible, you can have peacocks, snakes, and everything, rats, you name it, right? But they don't actually sell pangolin according to that menu anyways, because the pangolin trading is illegal. So maybe this is a legal sort of a price list, and then if you negotiate, you can get some illegal species. But civets, so these are the red boxed. Civets is traded, and civets you can buy the whole civet live, or you can buy civet meat. And as I said, civet play a very important role in the transmission of SARS. So I still think we should do more studies on the civet, its role in transmitting the COVID-19 virus. The reason I want to say that it's too early to conclude, and also that to identify intermediate hosts in the reservoir, it's not an easy task. So I'm going to review some of the stories or the experience that during SARS investigation, right? So this is the paper published in SARS in May 2003 to indicate that the SARS virus is present in live animal market, just like the one in Wuhan. So this is in Shenzhen, so that you have these animals not only positive for the virus, but also positive have antibodies. Okay, so civets are the intermediate transmitting hosts from this paper. Then I led an international consortium with 5 institute scientists from Australia, China, and the USA, and we identified bats as the natural reservoir of SARS-related coronavirus. But in that science paper, which published in 2005, we only discovered a sequence which is 92% identical to the SARS coronavirus. So remember I said now the Wuhan Institute, my former student, they already identified a virus which is 96% identical to the COVID-19. In 2003, we identified a bat virus which was 92. And that virus was not able to use, we cannot isolate the live virus. And the virus cannot use the human ACE2 as a receptor for entry. Credit to my Chinese collaborators, we work together and they keep sampling in the same cave. They go there in Yunnan province and they sample this like 3 times a year. And then 8 years later, we finally isolate a live virus, that's the bat SARS-like coronavirus, and that now can use ACE2. So, of course, the technology, everything is more advanced than 17 years ago, but I always say, for the audience, in this seminar and for the general public, so be patient, give us some time, because this kind of science is not easy and we cannot really finish, right, in short time. So this is like, you need 10 years of intensive research. As I said, in the Lancet commentary, I talk about a difficult to name a virus, 100 years ago, if you discover a virus, you can name anything you want. You can name your scientist's name. I can call myself a Lymphasis virus or Wuhan virus, and we can call patient, like the cell line we use very famously, the HeLa cell, right? It's from a cancer patient and her name is HeLa. In this day and age, of course, you cannot do that. And so it's a really painful process because, scientists and the WHO and the national bodies, we just could not really agree on the name, So the Hendra virus in Australia in '94 initially was called the equine mobility virus because initially people thought the sequence suggests it's very closely related to measles virus in the genus of mobility virus family paramyxovirus. I was the one, sequence the whole genome. Nowadays, you do a genome sequence in 2 days. In these days, you sequence the genome, takes half a year. So once I had the whole genome sequence, I said it's not a mobility virus. I changed this to Hendra virus. And the Nipah virus initially was kind of called Hendra-like because they cross-neutralized, and then the Malaysian scientists changed it to Nipah. SARS initially was called atypical pneumonia and changed to SARS. MERS now call MERS, but it was HCOV Erasmus Medical Center, the name created by Dutch scientists. And now we know that this COVID-19 initially was 2019 novel coronavirus. Another disease is COVID-19, and the virus is SARS coronavirus 2, which I don't like because when you have a SARS coronavirus 2, you mean you try to imply there's only 2 viruses, but actually we know that in bats there SARS coronavirus 1, 2, 3, 4, 5, 6, maybe to 30 already. So I think I don't like it, but it's being used. So maybe I say it's my fault because maybe I start all this sort of mess of naming viruses. So what's next? What we can do next, and I think what we're doing is now there's many things we're trying to do, and I think the role of serology, as I say, currently next-gen sequencing, PCR, and there's so much media discussion about the test, right? PCR test is not accurate. Why we get positive today, negative tomorrow, positive again, negative again, right? So what I argue to say is we need to have serology. We have to detect the antibody, and serology can play many, many roles, right? The diagnosis, contact tracing, epidemiology, herd immunity. How do you know we have herd immunity? Not only binding antibodies, we have to really detect neutralizing antibodies. And also the longevity of the protective immunity. For example, we just did a serology for SARS survivors from Singapore. 17 years later, we still demonstrate in their bodies, they still protect neutralizing antibodies. So this kind of data is very important. And also the last point is that the natural reservoir and a transmitting host, that serology can play a role, So we, in Singapore, I have been developing serology. I have now 6 different platforms of serology tests. We were the first in the world to use serology to do contact tracing. And for those of you who live in Singapore, you're familiar, in the early time, we had 2 major clusters, both are associated with the religious gathering. And we believe there's a link, but we could not find. And then credit to the MOH contact tracing group, they spent months finally to narrow down a couple which attended birth events, but they declared they were not sick. So use serology, we found this case 91 and case 83. both have neutralizing antibody months later, basically, So this is the first example of using serology to solve a very important missing link. Now, the other thing I think you all heard from media as well is that there's a lot of discussion about why children seem to be less susceptible, or at least susceptible to disease. Because any other respiratory virus, whether it's a flu or ISV or other rhinoviruses, children are equally or more susceptible. So what I have shown, this is a China CDC data a month ago. You can see on the left, again, for those who could not read the Chinese, I just reversed the image, that the age on top is the young from 0 to 4, and the bottom is over 100. So that's the natural distribution, and the blue is male and the pink is female. So that's the natural distribution of a population in China. And on the right, what you see is the COVID-19 cases. And it's incredible, it's a reverse kind of image. And so basically, people over the age of 60, your risk is like 1,000-fold over people under 20. Okay, so this is a very unique, for COVID-19, because for other respiratory infection, you don't see this. All right. Okay, so I'm going to finish up, as I said, because I've been studying bat-borne viruses, for 25 years. I got the nickname of Batman, and I always correct people that I initially was a bat virus man. Only recently I started study bat. So bats obviously carries, we have lots of bat species. Actually, 25% of all living mammals on Earth is a bat, and the species diversity is incredible, over 1,000. For humans, we have 6 billion people living on Earth. We all belong to one species, basically, different ethnic groups. But bats have over 1,000 different species. And they're incredible. They have long lifespan. A 7-gram bat can live up to 43 years, So as you know, for mammals, the lifespan is relative to body size. So if you use a 1 dog year is 7 human year, then a 20-gram bats, a 7-gram bats, one bat year, I said, would be 20, human years. So if you multiply that 43, you're almost 1,000 years old as a human. And bats are less prone to cancer. And we know now bats carries all sorts of viruses, but asymptomatically, they don't get disease. Even more sort of incredible is that during flight, their metabolic rate can go to 15 to 30-fold of their base rate, and heartbeats goes incredible, over 1,000, and body temperature can go up to 42 degrees. And they fly quite a few hours, 5 to 7 hours per night. If your human can do this, even if you can fly, use your current body and use your current immunology and your physiology, we will not last. We will die very, very quickly, because this is a very high stress kind of a biological process. So more than 10 years ago now, we tried to zoom in to look into where the difference is. So we did a whole comparative genomics. We compared the bat genome and with human and the mouse genome. We found the two really interesting at the genome-wide, that 2 pathways are under positive selection, means the bats had to perform better than human. One is they have enhanced DNA damage repair system. The other is they actually have a reduced inflammation activity. For those of you who are interested in inflammation, inflammation play a role in all the disease process, including COVID-19. So how to become an extremely healthy mammal? Bats are definitely healthy. They live long, don't suffer cancer, and they have virus but no disease, right? So I think, to me, it's clear now. We know that it's due to their very, very well-controlled defense versus tolerance balance. Okay, so they have examples of elevate innate defense, but more importantly, they have examples of dampen what I call is over defense. We virologists have a cliche to say very few viruses kill you, we kill ourselves. It's our immune response. So this is an animation to see that if you want to keep yourself health, you can keep a balance. If your immune defense is not enough, then you get cancer and infectious disease, and you cannot clear and you cannot survive. But if your immune system go over, then you have other diseases like autoimmune. But no biology system is perfect, human or bats, right? So what we need is we need to adjust. Even our sleep, our diet, our stress, like right now, home quarantine, no travel, It's all going to cause stress. So what our body need to address during that adjust process is going to like swing, right? It go a little bit over, a little bit under, and eventually get right. But you look at this, how human does and what bat does. Bat swings as well, but the range of swing is much smaller. If you translate this into scientific terms, and these are the defense genes. So the X, Y, Z are different defense genes, and on the left is bat, on the right is human and mouse. As you can see already, MOG means this is a cell culture condition, 37 degrees, lots of nutrient, they're happy. For bats, some of the genes already switch on. For human and mouse, usually you don't switch on the gene. And then when you see stress factors like chemical inducers, or inducer 1 is a chemical, inducer 2 is a Sendai virus. So if you put chemical pressure or virus pressure, your defense gene goes up. But for bats, the range is small, and for human and mouse, it's much bigger. Okay, so this is where the animation just try to say that bats is so healthy because they get the balance right most time, whereas for human, we sometimes overreact. I don't have the time to show you data, but just to show you 4 examples of enhanced immune defense or innate defense: DNA damage repair, interferon alpha gene, heat shock proteins, and some of the efflux pump on our membrane, like the ABC transporters. So in bats, they do better than us, even before they see enemies. But they don't over defense because they have other mechanisms to dampen. For example, it's an M2 mediated inflammation pathway. The whole pathway is deleted. And the other inflammasome activation is dampened. The STING molecule is very important now in cancer aging research. It's also dampened. And the graft versus host disease. This is a disease that if you do transplant, you do rejection. So when we put the bat immune system into a mouse, we create a bat mouse. And if you do that with a human immune system, your immune system will attack the mouse and cause GVHD, whereas the bat immune system can tolerate that. So this is a dampen of this. So we published this paper in Nature Microbiology, and they publish a commentary of our work about dampened, immune innate inflammation and how bats can balance. And bats are really, really special. So these are 6 different graphs. The first one, A, you can see the life expectancy, the life, longevity, lifespan is proportional to body size. I mean, this is for mammal, right? So human, we live longer, much longer than mice. Bats are small, like a mice, but they live quite long, in the middle, like 30, 40 years. And I don't have time to go through all the 6 graphs, just to demonstrate to say out of 6, only 1 that bats is on the curve. All the other 5, bats is off the curve. So bats are very special mammal. They're different from land mammal. And I think our hypothesis is because they need to do so to adapt to their flight. So in summary, bat has a much better immune sort of defense and tolerance balance. And that leads to a lot of things that we human can only dream of. You have a slow aging process, long lifespan, less prone to cancer. You can be infected with virus, no disease, less inflammatory disease, less autoimmune disease. And now we found they have less metabolic disease as well. So I think, bats, we should not think bats are bad. Bats are good. They can teach us lesson. So I'm going to finish it to say are bats good or bad? Some people thinking bats are evils. Western cultures mostly, through the vampire bats, blood-sucking bats. So bats in movies are depicted as the dark, evil side. But when you think of Batman, so to me, in Chinese, those you can't understand or read Chinese, bats is a fool, and same as fool in happiness or fortune, right? So I'm a Chinese, so I believe that, bats will be fool for humankind, and we can learn a lot from bats. And with that, I'd like to thank you for your attention. Okay, so I need to go give back these to, all right.
There's some questions from the floor.
Okay.
Yeah. So the first question is is from Yash. So her questions is asking relates to the recent research from India reflecting the role of mRNA in providing better immunity against India's SARS-CoV. Does the COVID-19 strain has already mutated in a 3 to 4 months time when it was first discovered in Wuhan and then traveled to India? Or is something actually relates to the genome of India?
No, I think the every virus mutates, and especially RNA virus. So for COVID-19, coronavirus is the largest single RNA genome on Earth, basically. So they have more chance to mutate. The good news is that so far the mutations are all what we call non sort of significant mutation. It does not change the virus. So the vaccines, whether it's mRNA-based, protein-based, or live vector delivery vaccine, so far the sequence suggests that all the vaccine will be effective.
Okay. The next question is, Christina wanted to ask how to confirm where the virus is originated from.
Sorry?
How do you confirm where the virus is originated from?
Oh, yeah, so this is a very tough question, because what I believe is that currently we found a 96% identical virus in bats in southern China, but that's not the virus. We must have a virus much closer to that. So that's what I say. We are using serology, because if you do PCR and sequencing, the window is very narrow. Like in humans, the virus stay for 2 weeks, and in most animals they'll be similar. So if you go to a population, you have to sample like 100 animals before you get a few positive PCR. But once you're infected, the antibody will stay there for a long, long time. As I said, the SARS patient in Singapore, we have evidence 17 years later, they still have antibody. So I always say we should use highly specific serologic test, which I'm developing now, is to survey animals in the region, not only in China, but outside China as well. Because if you believe pangolin is the intermediate host and transmitting the virus, it's interesting because the illegal trading of pangolin is incredible, and the ones that sold in Chinese market illegally are mostly imported. Not the Chinese pangolin, it's the Malayan pangolin. Okay, so I think we should expand the surveillance outside China as well. Thank you.
Okay, the next question, how many strains are there in the SARS-CoV-2 currently circulating? What's the molecular-based criteria for strain categorization in beta coronavirus? Is there any references?
Yeah, so this is a very good question, because at the moment, you can call a strain even if there's a 1 nucleotide difference, okay? So if you use this, so what we are doing now is all the beta coronaviruses that you isolate and you sequence, we give it a location, date. So for example, we have beta coronavirus 2020 Singapore 1, 2, 3, 4, 5, 6, 7, 8, 9, we're up to 30 already. So basically, we already have at least 1,000 different strains, all around the world. But I have to say that these are strains very, very close related, differ by 1 nucleotide, for example. And so collectively, the species, I think, is 1 and is stable. But the strains, I think we don't have to worry how many strains, because they are basically functionally as 1. Thanks.
Okay. The next question, how many mutated strains of COVID-19 are found till date?
Yeah, that's as I said, when you say mutated, if it's a 1 mutation is also a mutation, then we have maybe over 100 now. Yeah.
Okay. The next question, how long does immunity last after infection with COVID-19, and are they predictors or longer or shorter immunity?
Yeah, it's a good question. And as I said, I have been working in this very hard. So all we can say is like, in China, of course, they had the patient first. So if you were infected, like, as I said, December 1st, right? So, we're April 2nd, so basically you already have 4 months, and definitely all these patients have very strong antibodies. So this, I think, is very, very hard to predict or estimate how long that immunity will last for SARS-CoV-2 2. But because SARS-CoV-1 is the same species, right? We say these are all within the same species called the SARS-related coronavirus. And then if you go there and, we have the data, and I'm going to publish very soon, it's to say for SARS virus or the SARS coronavirus 1, and we have evidence now, even 17 years later, they still have protective immunity, means they have neutralizing antibody in their blood. Thank you.
Okay. The next question, is there any evidence to suggest that snakes are unlikely the intermediate host?
Yeah, so I think the data to suggest is that the coronavirus they discovered in snakes are so different from human, and we know that most of the coronavirus actually are very species specific. That's why we have so many bat coronavirus, and yet we don't have many that infect human, because they are species specific. So to cross species from bats to human is already difficult. And then imagine if you cross species from bats to snake to human, this is much more difficult, because snake is not even a mammal, So usually that, the animals, when you are evolutionarily more related, there's more likely to spill over, So that's why if we have lots of monkey virus, and if monkey have a chance to interact with human, then we are in much higher risk than bats. Bats are evolution not that related, and also that, we don't have a close contact. So based on this, what I'm saying is that it's unlikely snake to be the intermediate host.
Okay. The next question, do you have any, just a minute. Can we use bat serology to identify a vaccine? Would bats have antibodies against SARS?
I don't understand this question, because yes, of course, the bat has antibody against SARS, but that's how that can help us to develop a vaccine. Vaccine is we want to induce antibodies in humans. So I don't think these are related. Yes, bats have antibody to SARS, but we cannot use that to develop a vaccine.
Okay. The next one, can you comment on the stability of the virus genome? It is reported to have 8 strains so far.
As I said already, more than 8 strains, but they are stable, because when you say it's a different strain, it just means there's a 1 or 2 nucleotide difference, and this is out of 30,000. So to me, is that the genome is quite stable.
Okay. The next one is, why is finding animal X is so important?
Finding animal X is important for 2 reasons. One is to prevent future outbreak, right? If the animal X is widely traded, and very close contact with human, then the risk is going to be always there. So that's number 1. And number 2 is that, scientifically, that if we know what is the animal X, then we can even block maybe the back to X transmission. So we have 2 chances to do that, right? X to human and back to X. So this is why it's all for future preventions, So this is why after SARS outbreak, just like, the Nipah outbreak, we have to know where the virus is from, and then the pig, right? And how the pig gets from bats, and then to say in Malaysia at that time, they have this practice of multiple farming. So they have a pig farm and then have a fruit tree farm, and the fruit tree provided a shelter, shade for pigs. So then the bats were flying nighttime to eat the fruits, and the fruits were dropped to the pig pen, and the pig get infected, and the pig transmit to human. So these are very important to know reservoir and the animal X and how they transmit. And then, from there on, Malaysian government just banning, fruit farming and the pig farming. It's very effective. So far we have not had another Nipah outbreak. So this is just, I think it's pretty obvious that it's important to identify not only animal X, but how they transmit to X and from X to human.
Okay, thank you. The next one is, where are we now in the development of vaccine? Is there any updates?
Yeah, so I mean, there's maybe 100 groups now develop vaccine around the world, and there are at least 2 already reached human phase 1 clinical trial. One in USA, which is mRNA-based vaccine. One in China is adeno vector delivered vaccine, and there are many, many more kind of in the pipeline. And but I think I have to emphasize to the audience, up until now, we call this vaccine candidates, okay? A vaccine will become a vaccine only when they are licensed. And that process can take very long. So we're hopeful by end of 2020, we'll have a licensed vaccine.
Okay. The next question is, what is the status of serology testing in China and in other regions?
Yeah, so currently there are many companies. The last I checked in the WHO webpage is 50 companies claim they have tests, but none of them are endorsed by WHO, means that that has not been field validated. And so everybody is doing serology tests, and I myself have developed different platforms. And so I have one pattern, and we are going to try to work with commercial companies and to commercialize this. Yeah, so until a WHO conduct a large scale test, and maybe they select 1, 2, 3 for international use. We have not reached that yet, but inside China, outside China, a lot of people are doing tests, and this is what we call research use only test. So it's a laboratory based test, it's not clinic test.
Okay, the next one is, can I know why humans get the disease? Is it because they don't cook the meat that they bought?
Oh, from animal to human, you mean?
Yes.
Yeah, so the origin transmission. No, you don't. I mean, very few people will eat raw sort of pangolin or raw civet meat. I think the meat will be cooked. But why? Even when you go to market, right? All the live animals there. If you the market is contaminated, the fecal material, the animal's sort of saliva. So even if you touch the animal and try to have a check, or you may touch the cage of the animal, that's enough for transmit. And then if you buy the animal to home, and if the live animal or the meat, if it's contaminated, and during that process you will be transmitted. So I don't think that it's due to raw meat. I think eating raw meat would be crazy, right? Mostly transmitting is during process of the meat. Yeah.
Okay. There's another question. Instead of bats, there's also argument where COVID-19 was started in US and thereafter brought to Wuhan military war game. China is quite successful in controlling.
Sorry?
Yeah, China is quite successful in controlling the virus, and as unexpected, China seems to be using TCM as treatment since there's no Western treatment for cure right now. Should TCM be promoted more or research to this area?
Yeah, so there's two questions there. One is this so-called the virus may start from USA and they went to a military sports meeting and brought it to China. I personally feel like at the best this is a conspiracy theory, but I think scientifically there's no data whatsoever. And then the second question of China control well using TCM, again, this TCM, I personally believe in TCM, but I think in terms of treating coronavirus as a specific sort of treatment, it's like infectious disease. I always say, it's human to human variation is huge, right? Initially we say only people over 80, 70 will die, but now we have people, as young as 9 years old and can die. So the trend is there. The old people is more at risk. But if you have 100,000 people infected, and if you treat a few thousand of them with TCM, some of them will get better, right? So TCM may play a role, but I just say scientifically we don't have the statistics, right? If you treat with TCM, how many people recovered, but how many people didn't, so that's the data is missing. So I don't want to sort of discount the role of TCM, but I'm just thinking that TCM may be useful for some people, but it's not going to be useful for every COVID-19 patient.
Okay. All right, due to time constraint, we do have some other questions that we didn't get to answer yet. I will jot down and I will send all those questions to Prof. Wang and I'll email back to you guys. So I would like to thank all of you who joined us today. I hope that you find this session useful and beneficial. So before we end the session, I just wanted to briefly highlight the range of products and services that GenScript have for COVID-19 research. This includes primers and probes, qPCR assay, ELISA kit, plasmid and positive controls, proteins and antibodies. You can find out more information for the products and services on our website. So thank you guys for joining us and have a nice day.
Okay, all done.